Hey guys, we should get started. So it's a tremendous pleasure today to welcome back yet again Mike Levine. And I should say that I was really excited to be hosting Mike again until last night, when I learned that for my entire career, I've been laboring under misinformation thinking that Mike was a New Yorker. This was a good aspect of him. And instead, last night, I find out he's actually born in LA. So it's too late to send him back, but, you know.
And I don't want you guys to freak out. You know, LA Jews are just like New York Jews. We're just a little more housebroken, just a little more domesticated, that's all. No significant difference.
So in addition to being housebroken, Mike has had a truly amazing career trajectory and has made really seminal contributions to our understanding of both early embryonic development and transcriptional regulation. He started out, did his graduate work at Yale, and then went on to do like a microsecond of postdoc. He did a year of postdoc with Walter Gehring and another year with Jerry Rubin before starting out as an assistant professor at Columbia University. And he was at Columbia for a while, then moved, I think, to UCSD, and then finally to Berkeley, where he is now, where he's head of the division that includes genetics and developmental biology. And if my eyes worked in this light, I would tell you what the division name is, but I can't.
Close enough.
So Mike has won pretty much every award that one can win. He's a member of the National Academy of Sciences, and really he has made amazing contributions to our understanding, particularly in Drosophila, of the homeotic complexes and the pair-ruled complexes that pattern the early Drosophila embryo, and more generally to transcriptional regulatory mechanisms that function throughout eukaryotes. And he will be telling us some of that work today.
The usual stuff, yeah.
Stuff, except unfortunately only Drosophila. He does actually do some interesting work relevant to neural crest, but he's not going to talk about that today. Instead, he's going to talk about whole genome analysis of transcriptional precision in Drosophila. So welcome, Mike.
Thanks, Carol.
Even if you're not from New York.
Right, so as Carol implied in her kind introduction, yeah, I'm kind of an old guy. For almost 27 years, the lab has worked on the patterning of the early Drosophila embryo. And I'm not going to take you through a history of all that work. Instead, I'm going to focus on some of the whole genome studies we've been doing for the past 3 or 4 years. And most of these efforts focus on the dorsal ventral patterning of the Drosophila embryo, which is controlled by a sequence-specific transcription factor called Dorsal. This is related to mammalian NF-kappa B. And here you see a side view of a 2-hour Drosophila embryo stained with an antibody against the dorsal protein, which is distributed in a broad nuclear gradient with peak levels present in ventral regions and lower levels in more lateral regions. This gradient controls dorsal ventral patterning by regulating something like 50 target genes in a concentration-dependent fashion, and some examples of dorsal target genes are shown in this multiplex in situ hybridization done by Bill McInnes and colleagues. This embryo is just an hour older than the one I showed you on the previous slide, so the dorsal gradient gets right down to business in producing different thresholds of gene expression. The gene visualized in yellow is activated by the highest levels of the dorsal gradient, those visualized in blue, green, and red by progressively lower levels of the gradient. And one point I want to make before I just very briefly summarize the question of how the dorsal gradient produces these different thresholds of gene expression is to note this pattern of expression in yellow. This is the snail gene, the Drosophila snail gene, related to Carroll's snail and slug genes involved in neural crests and vertebrates. Snail works as a boundary repressor, which keeps off a lot of these lateral genes in the mesoderm. So the ventromost cells that express snail will invaginate to form the internal mesoderm during the next hour of embryogenesis. The low levels of dorsal gradient activate these genes blue, green, and red in the lateral neurogenic ectoderm, and these genes are kept off in the ventral mesoderm by the snail boundary repressor. So it's forming a boundary between the mesoderm and the neurogenic ectoderm. And it's also important for the subsequent invagination of the mesoderm. This summarizes the analysis of something like 30 different dorsal target enhancers. We see 3 basic threshold responses. Type 1, type 2, and type 3 type 1 target genes are activated by the highest levels of the dorsal gradient. And these enhancers tend to contain a disordered array of low affinity dorsal binding sites and low affinity binding sites for other transcriptional activators involved in dorsal ventral patterning, such as twist. And these binding sites work in more or less an additive fashion to give this very simple readout of the dorsal gradient, of the highest levels of the gradient. Now, response to the lower levels of the gradient involves a little more work, and you see that the type 2 genes are activated by intermediate levels of the gradient, type 3 by low levels of the gradient in the lateral regions which will form the neurogenic ectoderm, the future nerve cord of the fly. And these enhancers start showing some fixed arrangements of binding sites. And I just want to emphasize, not every binding site and all of the type 2 and type 3 enhancers are rigidly fixed throughout evolution, but at least a subset of the binding sites do show conserved organization. For example, in all 5 different type 2 enhancers that we've analyzed, you see closely linked dorsal and twist binding sites. This is an absolutely optimal dorsal binding site that allows lower levels of the gradient to activate gene expression. And this asymmetric twist site is pointing towards the dorsal site to foster cooperative interactions between these 2 proteins. Again, this arrangement is intended to allow fairly low levels of the dorsal gradient to reliably occupy the dorsal binding site and switch on gene expression. And then, as I mentioned in the previous slide, one of the type 1 target genes is the snail repressor. The type 2 and type 3 target genes contain snail repressor sites, which keeps the expression off where there are peak levels of the dorsal gradient in the presumptive mesoderm to create this boundary between mesoderm and neurogenic ectoderm. But the main point here is you need a little bit of regulatory grammar, fixed arrangement of binding sites for responses to low levels of the dorsal gradient to get these reliable lateral stripes of gene expression. Now, a few years ago, we collaborated with Julia Zeitlinger while she was a postdoc in Rick Young's lab at MIT to do a whole genome survey of dorsal target enhancers. And altogether, there are about 100 such enhancers scattered throughout the Drosophila genome. And these studies involve ChIP-ChIP and ChIP-seq assays using antibodies against dorsal and antibodies against 2 other transcription factors involved in dorsal ventral patterning, twist, and snail. And these studies led to the identification of so-called shadow enhancers, which I'll now spend a few minutes talking about. So here you see 2 of the genes in this whole genome survey that are regulated by the dorsal gradient, Brinker and SOG. Both of these are involved in BMP signaling. The ChIP-ChIP assays accurately identified the known enhancers which regulate Brinker and SOG. In the case of Brinker, it's a 5' enhancer. You see this cluster of binding sites located about 10 kilobases upstream of the Brinker transcription start site. That enhancer we identified through conventional gene fusion assays some years ago. And in the case of SOG, the ChIP-ChIP assays accurately identified the known intronic enhancer, which we also identified through conventional means some time ago. But unexpectedly, these assays led to the identification of additional clusters which regulate the genes. In the case of Brinker, a so-called shadow enhancer located within the intron of this neighboring ATG5 locus. And in the case of SOG, a shadow enhancer located about 25 kilobases upstream of the SOG transcription start site on the other side of this linked gene. We call these shadow enhancers because they're literally found in the shadows of the genes that they regulate. They're in places that we would not have expected there to be regulatory information for these genes. And so the question is, why 2 enhancers for the same pattern of expression? And I should say that these newly identified shadow enhancers produce patterns of expression very similar to those generated by the primary or proximal enhancers and seen for the endogenous genes. So here we have 2 enhancers separated by about 25 kilobases that give similar patterns of expression. And so to get at the question of why shadow enhancers, why separate enhancers for the same pattern of expression, I'm going to briefly summarize an analysis of the snail locus conducted by a graduate student in the lab, Mike Perry. And I should point out that as many as 1/2, 25 of the 50 dorsal target genes that are patterning the early fly embryo appear to contain shadow enhancers. So it's not just a few genes, it's many of the genes, maybe half, contain shadow enhancers. So here's the snail locus. As I mentioned before, snail is activated by peak levels of the dorsal gradient in the ventral mesoderm. It is controlled by a proximal or primary enhancer located immediately upstream of the snail transcription start site. And we identified this enhancer like 20 years ago. But the ChIP-ChIP assays identified a shadow enhancer within the intron of this neighboring TIM17 locus. And when the shadow enhancer is attached to a LAGZ reporter gene, it produces a pattern of expression very similar to that seen for the endogenous snail locus and the 5' primary enhancer. And so what Mike Perry did was to make a series of back transgenes, about 25 kilobases in length, that contains the entire snail and TIM17 loci and associated regulatory sequences. And then he made derivatives through recombineering of this back transgene that inactivate either the primary enhancer or the shadow enhancer, not shown here. And he did the experiment in a good way. He didn't simply delete the enhancers. He replaced critical regions of these enhancers with random sequence, which inactivate the enhancer activities. Then he used the different back transgenes to do genetic complementation experiments. And a couple of examples are shown here. So in the left panels, you see embryos that are homozygous for a deficiency of the snail locus, but they contain one copy of the back transgene with both enhancers intact, the primary enhancer and the shadow enhancer. This back transgene produces nice expression of snail, seen in red, and invagination of the mesoderm during gastrulation. There's similar rescue with back transgenes containing just one enhancer, either the shadow alone, in this case, or the primary alone. Here, too, you get expression of snail and invagination of the mesoderm. But this is at optimal culturing conditions of 22 degrees. When you start raising the temperature to 30, which is a stressful temperature for the fly, normal, healthy fly strains develop fine at 30, but it's a little stressful. At 30 degrees, the back transgenes containing a single enhancer start breaking down. And to quantify the breakdown, Mike replaced the snail transcription unit with a reporter gene, the yellow reporter gene, within these larger back transgenes. Now, yellow is important for pigmentation of Drosophila larvae and adults. It's not expressed in the early embryo. It's a fairly neutral gene. And it has a nice big juicy intron, which allows us to do in situ hybridizations to detect nascent transcripts, active de novo transcription, using quantitative confocal imaging methods that you'll see in a moment. The snail gene itself lacks introns like many of the genes that are involved in early embryonic patterning. The genes are small, they lack introns, they're quickly transcribed so they can get right down to business during the very rapid time frames of Drosophila embryogenesis. Everything I'm talking about takes place in a period of about 1 hour. And so we can't detect nascent transcription of the endogenous snail locus. We had to use this trick of replacing snail with yellow to do the quantitative measurements to ask, well, what happens when you delete either the primary or shadow enhancer at different temperatures? At the optimal culturing condition of 22 degrees, it doesn't matter if you have one or two enhancers. So here you're looking at the activation of de novo yellow transcripts in an early nuclear cleavage cycle 14 embryo within a few minutes after the gene is first switched on. And you can see that both of these back transgenes give nice, sharp, and uniform homogeneous patterns of transcriptional activation. So the loss of the enhancer doesn't cause any problem at 22 degrees. But at the elevated temperature of 30 degrees, you start seeing a breakdown in the pattern. Gaps in the pattern. Literally, you get nuclei where snail should have been on 10 minutes earlier, it's still not on. You get these long, persistent periods of no de novo transcription, and you get an irregular lateral border of snail expression. In contrast, the back transgenes that have both enhancers intact, rather than lacking either the primary or the shadow, when both enhancers are attacked, you continue to get a fairly decent snail transcription pattern. And statistically, these two patterns are not different from each other. So these kinds of experiments suggest that the shadow enhancer ensures that critical developmental control genes like snail are turned on in the right place, in the right time, in embryo after embryo, in large populations of embryos, even when those embryos are under stress, like elevated temperature, or genetic stress. For example, if we take away one copy of the dorsal gene, that shadow enhancer buffers the expression to give reliable transcription. And so the shadow enhancer, I believe, is a mechanism of canalization. Canalization is a term coined by Waddington about 60 years ago to describe the fact that populations of embryos develop just fine even when they're subject to minor variations in temperature or genetic background. Now, I mentioned that as many as 1/2 of the dorsal ventral patterning genes contain shadow enhancers. even more of the patterning genes involved in the segmentation process seem to have shadow enhancers. For example, just about all of the Bicoid target genes have shadows, and I just want to briefly touch on this. So here you're looking at an early embryo stained with an antibody against Bicoid. So at the same time that the dorsal gradient is distributed in that dorsal ventral pattern I showed you, you get this anterior to posterior Bicoid nuclear gradient with peak levels present at the anterior pole and lower levels in more posterior regions. And this Bicoid gradient is important for the differentiation of head structures and for initiating the segmentation gene network, which first and foremost depends on the regulation of target genes called gap genes. All the gap genes have shadow enhancers. And here I'm showing the three major gap genes, Hunchback, Krupel, and Knirps. And just to focus on Hunchback for a moment, Hunchback is transcribed in the anterior half of the embryo in response to high and intermediate levels of the Bicoid gradient. It's off in the posterior half of the embryo where there are insufficient levels of Bicoid to switch the gene on. This on-off pattern in Hunchback transcription was shown to depend on a proximal enhancer, the classical enhancer, identified over 20 years ago in the labs of Nusslein-Volhart and Gary Struhl. And this proximal enhancer straddles the proximal Hunchback promoter. Well, Mike Perry, using whole genome assays, identified a previously undetected shadow enhancer located about 5 kilobases upstream of this classical enhancer, upstream of the distal promoter, which isn't switched on until later in development. And despite the fact that it's unlinked from this classical enhancer, the newly identified shadow enhancer produces a pattern of expression very similar to that seen for the classical enhancer. So it's just what I talked about for snail, brinker, and sog. You have two separate enhancers, in this case separated by 5 kilobases, giving more or less the same pattern of expression. And in this case, the two enhancers work in a synergistic fashion to produce authentic patterns of transcription. So once again, this is looking at a series of back transgenes that have about 20 kilobases of genomic DNA encompassing the Hunchback locus and the flanking regulatory sequences. The Hunchback locus was replaced once again with yellow to do the detection of de novo transcripts. And you're looking at de novo transcription of these target transgenes within 30 minutes after the Bicoid gradient is first formed. Very early response. When you have just one enhancer or the other, in this case either the classical enhancer intact but the shadow knocked out, or conversely the shadow intact and the classical enhancer knocked out, you get kind of sporadic patterns of activation. And for example, in the case of the shadow enhancer alone, 24% of the nuclei, which should show de novo transcription, which do show de novo transcription of the endogenous gene, fail to show transcription of the yellow transgene with just the one enhancer. But when you have both enhancers intact, you get a much more homogeneous pattern of expression, and you get basically multiplicative action, multiplication of the two error rates for each enhancer alone. So in some way, the two enhancers, these pair of enhancers separated by 5 kilobases, work synergistically to give a sharp and homogeneous pattern of transcription. And we see this kind of enhancer synergy for the other gap genes as well. This kind of a dramatic example. This is the two Knirps enhancers. Knirps is regulated by two enhancers, one in the intron and one in the 5' flanking region. The intronic enhancer gives an abnormally broad pattern of expression. It's nice and sharp, reasonably homogeneous, but that's just not the endogenous Knirps pattern. It's too broad. The 5' enhancer gives the proper spatial limits. It gives a narrow pattern, but a highly stochastic pattern of transcriptional activation. Very erratic, lots of nuclei which show expression of the endogenous gene, don't show transcription with the 5' enhancer alone. When the two enhancers were combined in the same transgene, so you got the intronic enhancer in the 5' position, the distal enhancer placed downstream of the lac-Z reporter gene, the two enhancers are separated by 5 kilobases, you can see they work synergistically to give very homogeneous pattern of transcriptional activation with normal and sharp spatial limits. So in some way, the enhancers are working in a synergistic fashion to produce an authentic pattern of expression. And I found this result to be very reassuring because, and maybe some of you have had the same experience, if you're trying to take down a gene and you want to understand its expression, and you start identifying regulatory sequences to mimic the expression of the endogenous pattern, we find oftentimes that a single enhancer taken out of a complex locus gives a nice pattern, but it's not quite right. Like the centronic enhancer of Knirps. It's close to the Knirps pattern, but it just isn't correct. And on its own, that's an abnormal, that's an aberrant pattern of expression. But it nonetheless contributes to a perfectly normal homogeneous pattern when combined with the other 5' enhancer. So you can find enhancers that don't seem to be doing the job when you analyze a given gene, but that doesn't mean they aren't contributing in a meaningful way to the authentic pattern. That's what this result suggests. Now, we don't understand the basis for this so-called enhancer synergy, where you take two enhancers, each of which isn't quite right on its own, combine them to get a really sharp and homogeneous pattern of expression. But this enhancer synergy is different from the kinds of enhancer interactions we've seen in the past. So this shows an old experiment from 1994 done by a former graduate student, Susan Gray, who took a minimal lateral stripe enhancer, the NEE from the rhomboid gene, and placed it just downstream of the minimal E stripe 3 enhancer, and you see an additive crisscross pattern of gene expression. So there's no synergy, there's no interference. The two enhancers work in a completely additive fashion. And for the most part, this is what we see. For the most part, when you take enhancers and start combining them in a common regulatory region, you get additive expression. And it explains, for example, 7 stripes of Eve expression produced from 5 separate enhancers, 2 in the 5' region, 3 in the 3' regulatory region. Sometimes in doing these experiments, and we've done a lot of these kinds of experiments over the years, you see enhancer interference. And this is an example that was documented by former postdoc Angela Stathopoulos when she was in the lab about 5 or 6 years ago. And here she did a very similar experiment to the one you see on the left. The Eve stripe 3 enhancer in this case was placed next to a different lateral stripe enhancer from the IND locus. And now the repressors bound in one enhancer interfere with the activators in the other. So for example, a variety of repressors keep the IND enhancer off in ventral regions. These repressors now interfere with the ventral expression of the Eve stripe 3 enhancer. And similarly, the Knirps repressor forms the posterior border of the Eve stripe 3 pattern, and Knirps, which doesn't affect the NEE in this fusion gene, causes dominant repression of the IND enhancer in this one. Can't explain how that works. I don't know why. I don't know why in this experiment the enhancers are additive, and in that experiment they interfere with one another. And in this third example I showed you of enhancer synergy, how the two enhancers actually help each other to give a correct pattern of expression. So we're seeing at least 3 different outcomes of combining separate enhancers in synthetic complex regulatory regions. This last case of the shadow enhancers is an example of synergy to give proper patterns of expression. So just to summarize the bit about shadow enhancers, I showed you evidence that they may represent a mechanism of canalization, making sure that critical patterning genes are expressed just in the right place, just in the right time, in populations of embryos exposed to raw nature, to the wild. And then I showed you cases where multiple enhancers, the pairs of primary and shadow enhancers, work in some unexplained synergistic fashion to give you very precise and sharp and homogeneous patterns of transcription. So I'm now going to switch over to the second sort of unexpected finding of our whole genome analysis of the pattern of the early Drosophila embryo, and that's paused RNA polymerase. So the shadow enhancers were a bit of a surprise because, we didn't think to look within the introns and neighboring genes to find additional enhancers. That could only come from a whole genome analysis. And the other surprise is paused polymerase. So this shows an example, a couple of examples of the results of a whole genome survey, chip-chip assays, using antibodies against RNA polymerase II, and looking at their distribution in the early Drosophila embryo. And again, these experiments were initially done in collaboration with Julia Zeitlinger, then at MIT. This shows 2 dorsal target genes, tail-up and rhomboid. Tail-up is important for the patterning of the dorsal ectoderm. Rhomboid, I've already mentioned, is expressed in the lateral neurogenic ectoderm. In early embryos, before the two genes are turned on, you see that they're really loaded up with RNA polymerase. The promoter regions contain large amounts of RNA polymerase. Now, we don't know if that polymerase is active or inactive. It's possible that the polymerase is bouncing on and off the promoter. We're just looking at average occupancy. It's possible that the polymerase never engages the DNA template. It may bind and fall right off, but we still see these peaks because we're averaging over a fairly long 2-hour time window. To try to distinguish whether this is an active or inactive form of polymerase, and I should just say this term stalled polymerase, I'm in a constant state of nomenclatorial agitation on some of these transcriptional terms, because not even the people who work in the very specialized field of RNA polymerase elongation agree on the use of terms that they themselves have concocted. As I understand stalled polymerase, this is a generic term that simply means the polymerase has access to the promoter before the gene is switched on. Before you get productive expression of the gene, polymerase nonetheless has access to the promoter. You don't know if that polymerase is a pre-initiation complex. You don't know if it's phosphorylated. You don't know if it melted the DNA template. You know nothing about it. You know it just has access. That's stalling. Pause polymerase is a very specialized form of stalling where the polymerase is fully active. It has engaged the DNA template. It has melted the promoter region and has begun transcription. And to test to see if that stalled polymerase might correspond to pause polymerase, we teamed up with Leighton Core, a postdoc in John Liss's lab at Cornell, to do Gro-Seq assays, genome-wide nuclear run-on assays. And the basic experiment's a miracle to me that it ever works because it's got like 85,000 steps. This is a gross simplification of what they do. We give them nuclei from early fly embryos, either wild-type embryos or different kinds of mutants. They take the nuclei and they treat them with biotinylated uracil, indicated by these purple diamonds. And they do the incubation and polymerase elongation of the intact nuclei under conditions where there is no new binding of polymerase. They remarkably add this sarcocele, this detergent, to prevent additional binding of polymerase. So all you get is elongation of fully active and engaged polymerase that's already there. They do this labeling very briefly. It's like less than a minute. They allow elongation to proceed for just 50 to 100 nucleotides. And by the way, that star, that sun, is the 5' cap. And then they isolate these small elongated RNAs from the gamish using the antibodies against biotin, and then do deep sequencing. And basically find that wherever we see peaks of polymerase binding based on ChIP-ChIP or ChIP-seq assays, straightforward polymerase binding assays, we see peaks based on the nuclear run-on assays. In other words, that stalled polymerase is truly pause polymerase. That polymerase is bound, it's engaged, it's fully active, it's ready to go. And we know much more now. Basically, on average, the polymerase binds to positions plus 30 to plus 50 downstream of the transcription start site. It has capped, the 5' cap modification. The polymerase is already phosphorylated on serine 5, but not serine 2. It's basically just awaiting one last step to be released from promoter proximal regions and productively transcribe the gene. It's a much easier way to think about gene activation and development than having to worry about 50 subunits of mediator and schmediator and TF2D and everything else. It really looks like most of the genes that we're interested in the early fly embryo are subject to regulation by Pol II elongation. I should say that about 10% of the genes in the Drosophila genome in the early embryo show pause polymerase. And these genes are greatly overrepresented by developmental control genes. So most of the genes you care about, Hox genes, genes that constitute components of Notch, Wnt, hedgehog signaling, these kinds of genes are mostly paused. Over 2/3, maybe 80% of these genes show pause polymerase in the early Drosophila embryo prior to their activation later in development. And so why? Why this particular strategy of gene activation for the movers and shakers of embryonic patterning? Well, prior to 2006, there are just a handful of genes that were known to be regulated by Pol II elongation and pause polymerase. The heat shock genes, which Rick could tell you about, were known for the past 20 years to contain pause polymerase. And there, it made sense because heat shock is a stress response. And what the pause polymerase does is to get you ready for rapid induction. And that's a do or die response. Either the fly turns those genes on or it's dead when you heat them up. And so having polymerase there was shown by Liss and others to accelerate the induction of those critical genes. And it's possible to think about developmental control genes in the same way. To put them on a hair trigger, they have to respond accurately and quantitatively to very transient signals, like localized FGF signals that come and go, and maybe by having the pause polymerase, they're ready to go and will reliably respond to these kinds of dynamic and transient signals in development. And I think that's essentially right, but a biophysics graduate student in the lab, Alistair Boddicker, also suggests that the pause polymerase is a mechanism for suppressing transcriptional noise in development. And so his thinking is summarized here. This shows kind of a textbook view of gene activation. You start with a naked promoter, and then activators like Dorsal bind to enhancers, maybe shadow enhancers, and then lead to the recruitment of polymerase to the promoter. And once polymerase is bound, you're off to the races, the gene is on. Now, the problem with this mechanism of polymerase recruitment as the key rate-limiting step in transcriptional activation is that it's really quite intrinsically noisy. Single molecule studies suggest that only 1 in 10 or even just 1 in 100 RNA polymerase binding events lead to productive transcription. Most of the time, polymerase binds to the promoter and falls right back off without having made any kind of primary transcript. Now, what that stochastic variation means is that if you consider 2 cells within a developing field in an embryo, even if these 2 cells receive the same inducing signal at exactly the same moment, they're not going to switch the gene on at the same time because of this inherent variation in polymerase binding and recycling. In contrast, if you're dealing with the pause polymerase, as I mentioned, the polymerase has engaged the DNA template, it has melted the DNA, it has undergone so-called promoter escape. It's made a transcript of 30 to 50 nucleotides. It's phosphorylated on serine 5. It is ready to go. And so now, when 2 cells with the pause polymerase receive that signal at the same time, they're going to switch the gene on at the same moment. Okay. So it's a cleaner, more reliable means of getting uniform patterns of de novo transcription. And to test this idea, Alistair examined the early de novo transcription of a variety of patterning genes. And he seems to see a correlation. We don't know causality yet, but he sees a correlation between pause polymerase and synchronous patterns of transcriptional activation. So here you see 2 genes, SOG and FSB, both important embryonic patterning genes. SOG involved in BMP signaling, as I mentioned. FSB is an FGF8 signaling molecule. SOG contains pause polymerase, FSB does not. And what you're looking at are intronic probes that detect nascent transcripts literally within 2 to 3 minutes after these genes are first switched on. And within 2 to 3 minutes of activation, over 90% of the nuclei, which will eventually express the SOG gene, show de novo transcripts. Quite synchronous activation. In contrast, in the case of FSB, which lacks the pause polymerase, within the first several minutes, only about 50% of the nuclei, which will eventually express the gene, show de novo transcripts, and it'll take another 20 to 30 minutes before this pattern looks like that pattern and all these gaps fill in. 20 to 30 minutes may not sound like a lot to you, but in the fly embryo, that's a lot of time. When you go from totipotent nuclei at the onset of nuclear cleavage cycle 14 to completely determined and committed cell types at the end of that process, just 1 hour or even 50 minutes later. And so now we're testing to see if we take synchronous genes with pause polymerase and make them come on in a stochastic fashion, does this cause developmental errors? And even more interestingly, are there genes that you want to come on in a stochastic fashion so you can have variable outcome, different kinds of descendant cells, and make these genes come on in a synchronous fashion and see if that causes any mistakes in development? These are very involved experiments, and I don't know if I'm still going to be alive when, the data actually come out, but we're actively pursuing that. But the main point here is that genes that have the pause polymerase appear to come out in a synchronous fashion, kind of as predicted from theoretical studies. And transcriptional synchrony may just be a means for tightening up coordinate gene control among groups of cells that are developing together. I just want to spend the last few minutes talking about my favorite topic of transcriptional repression. And as I'm very fond of pointing out, and you guys probably heard me point this out, and I don't know when the statute of limitations expire, but I still blame my mother for everything. It's pathetic. I'm 56 years old, but if you knew my mother, you would understand why I have an intuitive sense for repression, oppression, inhibition, suppression. It just came naturally from my childhood. And I can't get enough of repression. I don't know. It's kind of sad. All right. And so we're going to talk about different modes of repression, for the real aficionados. We're going to talk about this state of repressed but poised, and I'm going to contrast that with the state of repression called silencing. And I'll try to distinguish this and what it means. So here you see the status of the rhomboid gene in a mutant fly embryo where it is off. Rhomboid, I've mentioned before, is activated by low levels of the dorsal gradient in the lateral neurogenic ectoderm. It is kept off in the ventral mesoderm by the snail repressor. In this mutant, you only make mesoderm. So this mutant is called Tol-10B. It has high levels of the dorsal protein everywhere and high levels of the snail repressor everywhere. So here you see the 5' enhancer, which regulates rhomboid expression. It's located about 2 kilobases upstream of the transcription start site. There does not seem to be a shadow enhancer controlling rhomboid expression. This enhancer has binding of the dorsal and twist activators, but also high levels of snail binding, because as I said, in this mutant background, snail is everywhere. It's normally restricted to the ventral mesoderm, but in Tol-10B mutants, it's everywhere. And we have evidence that the binding of snail to this 5' enhancer prevents the ability of this enhancer to loop to the promoter. Okay, so it's kind of an anti-looping mechanism for short-range repression mediated by snail. But regardless of the mechanism, the main point here, snail somehow keeps rhomboid off in this genetic background. Despite the fact that it's off, you see plenty of pause polymerase in the promoter region. And I see this pause polymerase as a manifestation of developmental plasticity. We're looking at an early mesodermal cell that's not yet fully committed. Yes, this is a neurogenic gene. It normally doesn't come out in the mesoderm, but this mesoderm isn't that far along. It wouldn't take much in principle to turn rhomboid on in this fairly naive mesoderm, particularly because it has this pause polymerase. So we refer to this gene as repressed but poised. This is a term first used to describe a number of mammalian progenitor cells like stem cells. Rick Young's lab and others have found that a number of genes that are off in stem cells but will come on in some of their differentiated derivatives, that those genes have pause polymerase and are also repressed but poised. They're off, but they're ready to go. They're on this hair trigger that I've already mentioned. This repressed but poised state is distinct from silent genes, which completely lack pause polymerase and are therefore farther away from being switched on. And this idea of silencing comes from looking at Polycomb mutants. And so here you're looking at the epidermis secreted by advanced staged wild-type embryos and a mutant embryo called ESC. Everyone gets always hopeful about that ESC, but it's not embryonic stem cell. It is extra-sex combs. Never do I manage to do anything of actual interest or value or use to anybody, but in any event. So it used to be an obscure, homeotic mutant studied by Gary Struhl literally 30 years ago. And the ESC mutants show the same phenotype that you see for a number, 20 different Polycomb mutants, and that is the anterior body segments, that includes the anterior abdomen, thorax, and even the head segments, are transformed into the morphology of the posterior-most 8th abdominal segment. And this wholesale transformation of the anterior segments into the A8 identity is due mainly to the misexpression of a Hox repressor called ABDB. ABDB is normally transcribed at the highest levels in the posteriormost regions of the embryo, where it's important for the morphogenesis of A8, but in polycomb mutants such as ESC, ABDB is transcribed high throughout the embryo, and this ectopic transcription of the ABDB repressor leads to the inactivation of anterior Hox genes. So, for example, the antennapedia gene, which is transcribed in anterior regions in the thorax, where it's important for, among other things, the morphogenesis of the middle thoracic segment, T2, that gene gets switched off by ectopic ABDB products in the ESC mutant. And that helps transform T2 into an additional A8 segment. So a postdoc in the lab, Vivek Chopra, did whole genome ChIP and Gro-Seq assays on these ESC mutants. And of the approximately 20 polycomb mutants, he selected ESC for this analysis because it's the only one that shows maternal inheritance. And without going into the gruesome genetics, I will simply say, because of the maternal inheritance, we're able to get large populations of homogeneous mutant tissue. We don't have that 1 out of 4 problem that you get with a typical mutation like in all the other genes. So we can get homogeneous material in large quantities. Sadly, you still need large quantities to do these kinds of whole genome assays. And what Vivek found is that all the anterior Hox genes, which we know to be repressed in this mutant, due to the ectopic transcription of ABDB, instead of containing less pause polymerase, they contain more pause polymerase, which was really unexpected. So here's an example. This is the proximal-most antennapedia promoter. Advanced stage wild-type embryos show a little bit of pause polymerase. This gene is just transcribed in the thoracic regions of the developing embryo. In most tissues, there is no transcription, and the pausing you see here reflects probably that expression in the thoracic zone. In ESC mutants, antennapedia is shut off, or it's attenuated by at least a factor of 30 or 40 or more. And despite the suppression, we expected in the ESC mutants to lose this pause polymerase because the gene's getting switched off. But instead, you see a 3-fold increase in the amount of pause polymerase at the promoter. And this trend is seen for all the anterior Hox genes. Genes that we know are repressed, that we expected to lose their pause polymerase because they're off and they're not going to be used, instead show more pause polymerase. That seemed kind of curious. This is for any members of the Whittam lab who might be in attendance. Vivek forced me to allow him to do a micrococcal nuclease digestion. I don't know, it has something to do with nucleosomes. I think it's a DNA thing. Anyway, so this shows the same antennapedia proximal promoter region, the increase in the amount of pause polymerase in ESC mutants versus wild type, and this increase seems to correlate with diminished nucleosomes at the core promoter in ESC mutants versus wild type. And this is a consistent trend. In ESC mutants, there's a loss of nucleosomes at the promoter and an increase in the amount of pause polymerase. And this trend, increased pause polymerase, a loss of position nucleosomes at the promoter, is not just seen for anterior Hox genes that are repressed by ABDB, it's seen for like 2,000 genes in the Drosophila genome. It's a very clear trend that in ESC mutants, there's increased pause polymerase. Here's one dramatic example. This is a gene called WIT, or wishful thinking. It encodes a BMP receptor that's expressed in just a subset of neurons in the nervous system of advanced staged embryos. Now, this is a little tricky because you have to just think backwards here. So this is a wild type advanced staged embryo. 99.9% of the tissue that we grind up in these advanced staged embryos do not transcribe WIT. And so you don't see any polymerase binding because the gene is predominantly silent throughout the wild type embryo. It's just on in a few cells, we can't see it. Too small a percentage of the total mass. In ESC mutants, you get this huge peak of polymerase binding at the promoter, no change in expression. continues to be on in just a few neurons. This does not correlate with some massive de-repression of expression. The gene continues to be on in just a few neurons, but you see a clear-cut change from basically linear no polymerase binding to now a huge amount of pause polymerase. So I think is happening, and you got to be a repression nut to follow me, is that in wild type, the gene is silenced. You have the Polycomb repressors working, and what they do is to silence that gene in the vast majority of the tissues where it's inactive, and so there's no pause polymerase. When you take away Polycomb function in the ESC mutant, now you convert the silent gene into a gene that's repressed but poised. It's still off, but it's at a closer state of readiness for activation than the silent state. From silencing to repressed but poised, from repression to repression in the Polycomb mutant, but I think that the gene is now easier to induce, at least this is the idea, in the ESC mutants. But the main point here is that Polycomb seems to work, at least in part, by inhibiting polymerase binding to the promoter. And so I'm just going to summarize that. I mean, this is an oversimplified view, but I know this stuff at least is confusing for me. So this is what I think Polycomb does. I think it maintains the terminal identity of cell types by silencing inappropriate genes. So think about muscle genes in a neuron. Those genes are never turned on in the neuron, never will be turned on in the neuron, and so what Polycomb does is to silence those genes in this cell type where the genes were never on and will never come on. And that's what it does. And that helps maintain the stability of this identity. Now in Polycomb mutants, all of a sudden these muscle genes now acquire pause polymerase in the neurons. The genes are still off, it's still a neuron. Muscle genes aren't expressed there, but they now have the pause polymerase. And this may be a manifestation of restoring some developmental plasticity, going from a stable, silent, terminal identity cell type to one where it might be easier to flip the identity of that cell because a lot of unwanted genes that are normally silent, normally completely lack pause polymerase, now all of a sudden contain pause polymerase. So we're testing this simple idea. The simple model here is that Polycomb just tightens up the repressed state. It maintains cellular stability by keeping unwanted genes really off, by not even allowing polymerase to bind. You take away Polycomb, it doesn't mean you're necessarily changing all sorts of cell fates. Most of the genes are expressed just fine in Polycomb mutants versus wild type. But because many unwanted genes now have pause polymerase, we imagine that it's easier to flip the fate of those cells, to transform them into another cell type. And so we're looking to see if the Polycomb mutants are less stable, for example, in culture than the Polycomb plus, the wild type cells. But the main take-home message here is that Polycomb repressors work, at least in part, by inhibiting the pausing of polymerase at a number of developmental control promoters. So I just want to end by thanking the people who did the work I described. The first part on shadow enhancers and canalization in buffering changes in temperature, shadow enhancers working synergistically with primary enhancers to produce homogeneous and accurate limits of gene expression. That work was done by a graduate student, Mike Perry. The second bit on pause polymerase and synchronous patterns of transcriptional activation was done by another graduate student, a biophysics student in the lab, Alistair Boddicker. And then this last part on the Polycomb repressors, inhibiting gene expression through silencing by blocking pause polymerase, that was done by a postdoc, Vivek Chopra. And Julia Zeitlinger, when she was a postdoc in Rick Young's lab, laid a lot of the foundation of the studies I discussed today, and Julia now has her own lab at the nearby Stowers Institute in Kansas City. And I'll stop there and take your questions. Yeah, that's a really good question. I could tell you that ABDB is one of the few genes that shows misexpression in in Polycomb mutants like ESC mutants. And if you look at the pause polymerase, it has more pausing, than in wild type. Okay, so even though it's expressed in more cells, there's still plenty of cells where it's not expressed, where it's normally silent, but it nonetheless also behaves like all these other genes. It has the pause polymerase. Now, I think that getting the pause polymerase is step one for gene activation. Step two is having the right combination of activators present to release that polymerase and get meaningful expression. For whatever reason, ABDB has those activators present, but they're not present for the vast majority of the other genes that behave in this way. But even ABDB follows this rule that in Polycomb mutants, there's an increase in pause polymerase.
So, your model for the shadow enhancers suggests that they work kind of independently, but together. And so, but is that really true? Does the mutation of one shadow enhancer, like shadow or primary enhancer, affect the other enhancer in any way?
I really wish I knew exactly what I meant by enhancer synergy. I'm trying to use a vague term because we just don't know mechanistically, what does it mean? When you have, like, the Knirps enhancer gives a fat pattern and the 5' enhancer, which gives a narrow pattern, you put the two together and you get a homogeneous narrow pattern. What does that mean? Maybe it means that this enhancer is better at looping to the promoter, the narrow one, and that facilitates the subsequent looping of the fat enhancer so that it can only loop in the domain where the narrow enhancer does it. We just don't know. There's too many sort of possible mechanisms. And so is the looping of one enhancer really linked in some way, either positively or negatively, to the looping of another enhancer, or are they totally independent?
I don't know the complement of proteins that one enhancer is the same whether or not the shadow enhancer is present.
Right. So we just don't have sophisticated enough methods. We've tried using chromosome conformation capture assays. It's just not quantitative enough for us to be able to say, geez, when we remove the shadow, do we get impaired or augmented looping of the remaining primary enhancer? This is what we would like to know. So we're trying to visualize it. We're trying to use, we really have hopes of just visualizing enhancer promoter looping directly in the microscope. This is what we're investing heavily in.
Right. And you say homogeneous and active. Those are two different things, right?
Yes.
So not only do you get sort of a Swiss cheese effect within
Yes.
the Knirps band, but it's broad.
Yes, exactly. So the homogeneous expression could be explained through completely independent action. Okay, you don't have to invoke anything special there. If you get a 10% failure rate here and a 10% failure rate there, that's going to be inherently multiplicative. You're going to get a 1% failure rate if the two enhancers independently loop to the promoter. And to some extent, I think that's correct. But it doesn't explain the correct spatial limits. It gives you the homogeneous expression, but it doesn't obviously give you the correct spatial limits. These are pretty new results that I'm an old dog. I didn't think I would see new tricks when it came to combining multiple enhancers because it was our bread and butter for many years. And so this is kind of fun to think about this stuff again. I'm just going to work from front to back, if that's okay. Or should I work from back to front? Are you going to be, you want to be the maitre d'? Okay, thank you. You mean, but before I wasn't?
Yeah.
Okay.
So many of these shadow enhancers are buried in the introns, bits and pieces of adjacent genes.
Yes.
Now, an example you had up there, you had snail right beside it was TM17B2.
Yes.
I'm going to bet that if you look at the expression pattern of TM17B2, it looks nothing like snail. Yet it's surrounded
You're correct.
on all sides by snail.
You're correct. Yes, you're correct. So why is it refractory to all these snail regulatory sequences? TIM17B2, beautiful name. I think that even the C. elegans people could appreciate that name. It just stands for trans-integral membrane protein number 7, right? Unknown function. It's not expressed in the early embryo. And so I don't know. Now, years ago, we found cases of enhancers that would preferentially go to one promoter and ignore another neighboring promoter. Sometimes that involves tethering sequences, which are right next to the promoter. So one possibility as to why these enhancers specifically target snail and ignore TIM17B2 is because immediately 5' of the core snail promoter is a kind of a tethering sequence, Velcro, which helps reel in those distal enhancers, so they'll jump right over TIM17B. The other possibility is sometimes the core promoter elements themselves will determine what enhancers they listen to and which ones they ignore. And there are two basic classes of enhancers, of promoters in Drosophila. TATA, or promoters that contain initiator plus the downstream promoter element characterized in Jim Katanaga's lab. And it's that second category, initiator plus the DPE. Those tend to be the pause promoters. The TATA-containing promoters tend not to be paused. It's not 100%, but that's the trend. And we know of cases where enhancers will preferentially go to one class or the other. Some enhancers just, for reasons we don't understand, prefer TATA-containing promoters. Other enhancers may prefer that pause promoter containing the initiator element and the DPE. And so I don't know if something like that is going on here. Certainly the core promoter sequences look, of the two classes I just described, with snail being an initiator DPE style promoter, TIM17B2 having a TATA. But I don't know if that, we have not done experiments to see if that's the basis for the differential targeting.
Does that hold true in stem cells and vertebrates too, or are they not
Say that one more time. You talk so fast, Carol. It's like, I don't know if you guys noticed this. I don't know if you guys have noticed this. But, I mean, it's so pathetic. I showed 25 slides in this talk. 10 years ago, it would have been like 50 slides. So it's like, every several years, I slough off another, because I'm talking slower, so I slough off another 5 slides. You have me back in 5 years, it'll be like a 1-slide seminar. And it's horrible. Anyway, so slow.
Stem cells, they've shown poised polymerase.
Yeah.
Are those TATA-containing?
Oh, geez, that I don't know. I don't know if, and let me be clear about pausing in Drosophila. In Drosophila, the pause promoters tend to lack TATA. They contain initiator, and they contain a specialized version of the Katanaga DP element that's very GC rich, that we call a pause button. But it's really a specialized DPE, and it's located at plus 30 downstream of the transcription start site. And that's where the polymerase is arrested. The last sequences it transcribes is the specialized DPE, the pause button, and then it arrests to await, its release by appropriate activators. I don't know if it's coming out that way in mammalian systems or not.
I have a question about your terminology.
Yeah.
Repression versus silence. To me, repression means that there's some active process keeping a gene off.
Yep.
Whereas your data, I think, argue that there's a default state of paused but inactive polymerase that is sort of what happens to genes that ought to be silenced but aren't being silenced. Whereas repression is happening presumably in other places in the genome where there is no paused polymerase because there's the repressor binding and keeping that.
Right.
So, am I
No, you're fine. I mean, first of all, I'd like you to spend a little time with my mother, because it seems to me that you just had too happy an upbringing, not to see these subtle shades of oppression, repression, suppression, inhibition, and so forth. No, but look, I hear what you're saying. So in the case of what I call repressed but poised, where you have the pause polymerase, there is a sequence-specific repressor engaged snail which is actively keeping that gene off. Okay? We do not believe it's keeping it off, however, through the pausing. I mean, it's formally possible, and this we call the Kornberg constipation model, not named after Roger Kornberg, because if he heard that term, he'd kill me, but his younger brother Tom, who suggested that, look, you can only load polymerase because of its Stokes radius. The Kornbergs know what a Stokes radius is. I had to look it up on Wikipedia, of course. And apparently the polymerase can only load even when you have maximum activation once every 75 nucleotides. That's it. That's like bumper to bumper on the freeway of the gene, once every 75 nucleotides. This pause polymerase is invariably located between plus 30 and plus 50 nucleotides downstream of the transcription start site, meaning that it will preclude the binding of a second polymerase. You will get orderly, just one polymerase at a time, in an orderly fashion coming through that promoter using the pause polymerase, because at every round of transcription, the polymerase staggers at the pause site for several seconds at least before it, even when the gene is fully on and goes on and transcribe. Okay, with that said, so it's possible that the part of the act of repression by a repressor like snail is holding that polymerase in place, and that through the constipation mechanism will prevent additional polymerases from coming on. And we can't rule that out. And nonetheless, I'm still calling that repressed but poised because having polymerase with serine 5, phosphorylation, that gene is still, kind of on a hair trigger. The second you do anything to remove snail in this case, it's off to the races. That gene is on. Now, I distinguish that from silencing where there's no pause polymerase, but that doesn't mean that there aren't other repressors also ensuring that that gene is kept off. Okay, there could very well be, and we know in some cases. There are, like, for example, if you think about Entanopedia, we know that an ABDB is bound to the promoter region, actively keeping it off.
I guess what I'm thinking is this sort of oversimplified idea that, let's say there are promoters where the default state is paused polymerase, and they require an activator to bring in PTEC B.
Yep.
And then there are promoters like, let's say, Autobox containing promoters that are
Yes.
NB.
Yes.
And are regulated at the level of Pol II recruitment.
Yes.
And so if you have the first class, it wouldn't actually require repression. It would require the absence of an activator.
Correct. That is correct. Absolutely correct. Yeah, you're 100% right. It is an oversimplification, just for the purposes of where we stand right now in our understanding, so we can.
Well, I think terms like repressor come from our original view of the promoter as a desert of polymerase, to which polymerase can either be actively recruited,
Yes.
or that can be prevented, and that would be repression.
Well, the repression I grew up with, with the lack. No, I'm not talking about this. I'm talking about Jacob, my father's, Jacob and Monod. That repression was precluding polymerase binding in the end. That's the classical bacterial mechanism for transcriptional repression. And that's, what I've always looked for.
One default state.
Yes.
You're saying there are two default states.
I'm saying there's two default. I think I would agree with basically what you're saying.
So this is a little jumbled of a question. So if you take,
As long as you talk slow, exactly.
and so if you think Polycomb is involved in plasticity, you would maybe predict that it wouldn't have shadow enhancers, so that in cases where you would need to, there would be more plasticity. Have you looked at that? Does that make sense?
It does kind of, in a strange way, it makes sense to me. And I just, I don't have an answer because we haven't really looked at the data set with that particular angle in mind. There's the general question regarding shadows. Do they make things more flexible or more hardwired? And I don't know which way to go on this. So in other words, I can imagine, shadow enhancers are a cheap way to get things right. You can duplicate enhancers. It could be all sorts of cryptic duplication events to give you these additional enhancers. I'm sure we're loaded with shadow enhancers. The junk is loaded with it. You don't particularly need it. So the question is, genes with these extra enhancers, are they more evolvable or less evolvable? Are the two enhancers working to ensure that you do it only in one way so that you never deviate from that one pattern of transcriptional activation? I would think that if you have the shadow, the other way to think about it, and I think this is sort of at the core of your question, is that by having the shadow, you have the opportunity to evolve new binding sites for this one enhancer, produce a new pattern of expression. Maybe there'll be a little cost of robustness, but at least you have the other enhancer there to ensure that the core function is dealt with. Whereas if you only have one enhancer, it seems to me that's a much more difficult situation to evolve. You start monkeying around with that enhancer evolving new binding sites, you may get into a lethality situation very quickly. So it all depends on which way it's going to go with the shadow enhancers. Are they facilitating change, or are they locking in, one way of doing things? And you can, from a population genetics point of view, you can argue both. Both arguments, make equal sense. So we just don't know.